Great. Okay, so I thought I would take this opportunity to tell you, as it says here, my journey from going from undergraduate in physics and a PhD in physics to becoming a neurobiology professor. And I also want to say that to do this in the context of this phenomenon called imposter syndrome, which is something that I and I think probably many people in this room struggle with. And so this gives me an opportunity to give you some context for this, and hopefully at the end of it I'll reveal my strategies for how I deal with it that will hopefully help all of you. Okay, so what is imposter syndrome? So imposter syndrome, of course we go to Wikipedia. I love Wikipedia, and it actually has a definition that explains the phenomenon perfectly. It's a concept describing high-achieving individuals who are marked by an inability to internalize their accomplishments and a persistent fear of being exposed as a fraud. It is particularly prominent in high-achieving women. And if you go further in the Wikipedia site and you look at the demographics, it's a phenomenon suffered by African Americans who feel like by being the beneficiary of affirmative action, they may not think that their abilities and their skills are what allowed them to be hired. It's commonly reported by graduate students, scientists beginning tenure track positions, basically everyone in this room. So, and so I think it is a real problem, and I think it's something that we all have to confront. But it turns out we're in good company. These are some people who have quoted as saying they suffer from imposter complex, and when you look on that World Wide Web, there's really a lot of sites about this, and a lot of people who suffer from it. And so I'm just picking the people I respect most greatly and was shocked to read. So Maya Angelou said that I've written 11 books, but each time I think, they're going to find out now, and that I've run a game on everybody. Tina Fey, who I think is fantastic, has had tremendous amount of success. Lisa, she struggles with it, but she admits that she handles it pretty well. There's Sonia Sotomayor, Sheryl Sandberg, Michelle Obama, Jodie Foster, you name it. Everyone suffers from it. And in fact, if you look at all of the faculty here in MCB, I would say that everyone I asked suffers from it except Dan Feldman, my husband, who I asked this morning at breakfast whether he suffers from it, and he said no. So, okay, maybe it's not universal, but it is a very common, and I think it's very common, although associated with women and minorities, I think it's very common to find men who suffer from it. So, let's just take a little show of hands. How many of you have felt that you have, that you can be characterized as suffering from imposter syndrome? There you go. So this is very topical for this audience. Okay, so let me go through my achievements and my journey and all my excuses about how I didn't deserve my successes. Okay, so I got my PhD in experimental solid-state physics. I studied liquid crystal molecules, and I used a technique that's called second harmonic generation. And basically the question was, that we were intrigued was, if you take liquid crystal molecules that are polar, they're essentially little dipole moments, and you put them on a polymer surface, can you get all those little dipoles to align? And so I did this work with Ron Shen. He is just recently retired as a member of the physics department here at Berkeley. And this is what my data look like. And so what you're looking at here is the fluorescence that comes from a second harmonic signal. So you would have your surface down here, you would shine a laser light on it, and you would look at the second harmonic light that's generated that's coming off it. So this is a nonlinear mixing process that's very sensitive to the orientation of molecules at the surface. And so what you see in the circles, the open circles, are what you expect when you have an isotropic distribution of these molecules. And then if you rub the polymer surface with a cloth, you put down these liquid crystal molecules, you'll find that they orient. And if you change the polarizers on the input and the output of the light, and you rotate your sample, you see these interesting figures that emerge that are shown in the black. So one will be example for those who are physicists of having what's called S polarization in and P polarization out, and just changing the polarizers on the input and output gives you these interesting shapes. Now the thing I'd like to point to here are these solid lines that connect it. That is a model that has one fitting parameter, which is the angle of which these molecules are oriented, and that fits all of the data. I miss those days. As I become a biologist, you can have this one very simple equation, and you can just get this beautiful description of understanding, and get a data point that you can really very firmly believe. Okay, but that all went away. In my first postdoc, it didn't fully go away, which I did was at Bell Laboratories. So at that time, Bell Laboratories was the research arm at, so I should say at the end of graduate school, I didn't really know what I wanted to do. There were too many physics PhDs for the jobs that were out there, and I explored a lot of different areas. And Bell Labs was an interesting opportunity because there I found this group of physicists led by David Tank who were doing neurobiology. And I didn't really know what neurobiology was. In fact, I definitely didn't know what neurobiology was. In fact, I'm pretty sure I don't really think I knew what a cell was. I had never taken a biology class since high school. It was all very new to me. And what we did, these were in the early days of calcium imaging, which is a technique that is used a lot in physiology research. And so I got to be there at the front end of this kind of development. And Bell Labs was a safe place to do this because it's where a lot of physicists went to do postdocs. So I did a postdoc there, and the experiment was to take the brain out of a frog and then look at the part of the brain where the retina sends its connections to the brain, and that's called the tectum. And then you could stimulate the optic nerve, like what would be all those inputs from the retina, and you could look at the change in calcium at the very tips of those retinal ganglion cell, those retinal axons, the wires at the ends of the wires, and look at how much calcium went up in those cells. And calcium is an important signal that neurobiologists care about. And so this is what the data looked like. You could measure the electrical response, which is shown in this sort of double-peaked curve here. And then you could look at the change in the fluorescence of the dye that this brain's been soaked in that's very sensitive to calcium. And you get something that rises quickly and decays slowly. And so the point of these experiments was to see how we could use data with this sort of kinetic parameters and see if we can learn something about the concentration of calcium in these neurons. And in order to do this, we were very interested in how is calcium processed in a neuron. And so we would do experiments where we'd put on a drug and you would do a rapid succession, 3 quick excitations to that optic nerve, and you'd get these responses. And then you would put on a drug that buffers the calcium and look and see how those responses changed. And this is when we weren't, in order to make sense of this data, in order to have some context for it, we built models that ran simulations. And so we used simulations to test this hypothesis, and this is sort of what we were able to do was to say this is what we would expect given our model of how we think calcium, the concentration of calcium is influenced in control conditions versus in EGTA. And then we could further say, well, this is the fluorescence trace. What we really care about is what actually calcium is doing, and we can further go back with our models and sort of estimate what exactly are the calcium dynamics. So the point to come away from this is that simulations, at least when I do neurobiology research, are key for me to feel like I understand, what is the underlying physical process. Like, am I getting this right? And simulations is a way to sort of give you some context in which to think whether or not your results make sense. Okay, but then a lot of people ask me, why did I switch from physics to neurobiology? And it all happened because I read these papers that were by Hodgkin and Huxley. A lot of people, and we'll talk about that later if you want, but this is the most elegant research that's ever been done ever, and I was very excited to read about this when I was a physicist, and when I read about the work done by Hodgkin and Huxley, which was a way to explain this phenomenon called an action potential, I was convinced this is the kind of science I wanted to do. But anyone who's ever taken a physiology class from me has heard this lecture, and so I won't belabor it now. Okay, so for my second postdoc, so Hodgkin and Huxley convinced me I wanted to be a neurobiologist, but I hadn't yet really worked with a true neurobiologist. And so for my second postdoc, I decided to work with a real neurobiologist, and that was Carla Shatz. And that's when I came. She was a professor here at Berkeley at the time. And what we worked on there is this phenomenon called retinal waves. So what you're looking at here is a retina that has been isolated from a very young mouse. And what we were interested in is, are there neural signals in the developing brain before the animal can even see? So mice can't see when they're first born. They don't have the photosensitive cells in the retina are not yet mature, yet the retinas are not silent. They're spontaneously generating this activity pattern. And so this was an opportunity to use these skills I had in calcium imaging to actually address this fundamental question in neurobiology. And so this is a retina that you're looking at here. It's an individual neuron within, so the retina is the cup in the back of your eye. You can dissect it out, you can isolate it and have it lay flat. And this is about a quarter of the whole retina, and these individual little things that are below the resolution of this camera are neurons. And we can play a movie of what the calcium is telling us about the underlying neural activity. And you can see there are these spontaneous, what we call retinal waves. So there's no vision going on, but you can, but spontaneously you have these test patterns that are being generated in the retina. And these test patterns are something that I've actually spent my entire career trying to understand how they're generated and what role they play in setting up connections in the brain. But at the time when I was studying these problems, and I should say I've shown this movie a lot, and I like to show this movie because these are actually the first 4 waves I ever recorded after I set up the microscope in Carla Shatz's lab, and so they held a special place in my heart, these 4 waves. And I spent a lot of time analyzing them. So these are these 4 waves again, and this is actually when Adobe Photoshop was first developed, and I learned how to use Adobe Photoshop, and I had to extract my data off of videotape. I had to do frame captures, and I had to import it into Adobe Photoshop, which was a nightmare of a program, and we barely had computers that could run it. And I made these pseudo-color summaries of what the propagation properties were of these waves. So here was the first wave, here's the second wave, and each sort of colored landmass is a different wave, and we were analyzing sort of their propagation properties using this analysis. And again, we turned to using simulations to see if we were right. So our goal was to understand what is the circuit that generates these waves. And like I said, we've spent 15 years trying to understand all the cells and how they're connected. And actually, a lot of this work came to culmination in work done by Kevin Ford, who is a graduate student in the MCB program, who graduated a few years ago. And this is just an example of what Kevin did, is he built a kind of model network of the cells that we thought were very important. He assigned them to have certain electrical properties, and then he ran simulations based on this network and to see how well these simulated waves would mimic the properties of the waves that we actually recorded. And these are just examples where the blue is the simulated waves and the white is based on the calcium imaging, and he found that the frequency of the waves, the speed, and even the distribution of their sizes is something he could recapitulate. And from that, I could conclude, I think we understand the circuit, right, to generate these waves, because we can now model all of the things that we do know and recapitulate what we see experimentally. Okay, so I told you sort of a little bit about the science, but what were my excuses? All right, so I got a PhD in experimental solid-state physics and optics. You know, I had very few people in my family who had even graduated from college, let alone gone on for a PhD. You know, the whole thing seemed ridiculous when I was doing it. When I was majoring in physics, I didn't even tell my family because I thought they would think I was being ridiculous, because they're, you know, who did I think I was to do that? And I figured if I stayed around long enough, they couldn't really kick you out of graduate school. For those who are PhD students here know that you actually don't even have a thesis defense, right? So as long as you write your thesis, you know, you're going to get your PhD. And so I was in graduate school for 7 years, and they'll give it to you if you just sort of stay around long enough, not if you're smart enough. Okay, I went to Bell Laboratories. I actually had a fellowship during graduate school from AT&T Bell Laboratories, which meant that you had some sort of priority for a position there afterwards. And so they basically had no choice, since I was a fellow, and they had to hire me, and they regretted it the whole time I was there. My second postdoc, I figured, well, Carla Shatz really wanted someone who could understand the imaging. She needed, you know, a physicist to help her do it. You know, she clearly didn't do it because I was smart enough to understand the biology, but she must have done it because she needed someone who knew how to deal with a camera and a computer. So that was why she hired me. My first job I got was at the NIH, and I was actually hired by Story Landis, and she was the new head. She was a woman, and I figured that she really wanted to hire female faculty, and so that's why I got that position. Then we went to UC San Diego, and I figured that they hired me actually because they wanted to hire my husband. And you know, I have to say, I say that now, and I've said it a lot, and I don't even believe that, right? Like, I really believe they wanted to hire me, but I still say it. So that's, so at the time, and then I have to say, at Berkeley, Dan and I were both, Dan Feldman's my husband, we were both hired here, and several people had said no, and they were desperate to get, anyone, and so they hired both of us because they were just tired of searching and we were willing to come. All right. it's exhausting. And it's ridiculous, right? Did I not sound ridiculous? And that is how all of us sound when we are saying this in our own heads. And so the question is, how do we stop sounding so ridiculous? And so, when does this imposter complex go away? And let's see. So for me, it mostly goes away when the data is really interesting, right? I love doing experiments. When you're sitting there looking at data, it doesn't matter. No one else is judging you. It's just you and your data, and there's no imposter there because you're just looking at the laws of nature. And so I'm going to just give you just to give you a sense of this, I'm going to just tell you about a current problem which I like to think about in the lab, and it's a recent discovery. So just very quickly, the retina, we study these retinal waves. They're spontaneous. And when we say they're spontaneous, it's because, so just for those of you who don't think about the retina all the time, the retina is this layer of cells in the back of your eye. The output are these cells down here. They send their axons that go up to the brain. That's your optic nerve. And everything starts with these cells that are called photoreceptors that absorb photons and turn them into the electrical signals of the brain. Okay, in development, when there are retinal waves, these ganglion cells are there, but those photoreceptors, they barely work and they're not connected, right? They're completely not connected to the ganglion cells. Yet, in some experiments being done by Alex Trejak, who's a postdoc in the lab, who's here in the audience, he found that when you can be imaging and you can flash a light at this retina, which is there, and you can start a wave. And when that light's on, you can turn the light off, and that will also start a wave. You can see another starts a wave. What the hell, right? So the photoreceptors are not connected. Where's the photosensitivity coming from? How is this happening? And that's a mystery that we're trying to think about now. And when I'm thinking about that, I don't think about being an imposter. Okay, but there's other strategies that I've also used when the data is not always there. So the first one is, it goes away when I'm too exhausted to care. And I've told a few people this story. When I had a kid, and I was an assistant professor, and my grants weren't getting funded, I barely could find time in the day to pee, let alone worry about this, right? So I had no time to worry about this. I was just trying to get through the day. And at that point, I remember one of my colleagues, a young woman, came and talked to me, and she was talking about how she had just gotten some award and she didn't deserve it. And I just looked at her and I said, how do you have the time, right? How do you have the time to worry about this? And that's when I realized if it's something that's that expendable, like when I don't have time to worry about it, it goes away, then this can't possibly be real, right? It can't be worth the ATP. The second thing that makes it go away is when someone asserts that I'm an imposter. Okay, as soon as anyone tells me I don't deserve to be somewhere, then I get really pissed off. And then the whole imposter complex goes away. So that's another way that works for me. Then there's a case when I actually have been an imposter. And the time I have been an imposter is when I had no training in biology. The first class I had to teach was human physiology. And when I stood up and gave 4 lectures on the kidney, I was totally an imposter, right? I didn't know anything about how kidney worked, and here I was, teaching 300 undergraduates about how it worked. And you know what? It's not that bad, right? So I knew more than they did. I had read the chapter before. They learned something. So, being an imposter, it's not that bad. Still, even if you are an imposter, you're still probably evoking knowledge in some way. You're helping people learn things. And then the other thing I would recommend is reading this article by Indira Raman, which was published in eLife. She writes beautifully. So first of all, she's a wonderful writer. And she wrote this article that's called The Truth is in the Distribution. And this is actually Indira here. She's a professor at Northwestern University and a great physiologist. And she addresses many aspects about being a woman in science, but one of them is this feeling that whenever you get asked to do something, are you getting asked to do it because you're a woman? And she said, or are you getting an award because you're a woman? And she said, that may be, and there's a chance that that's the case. And, when you walk on the stage, you might walk on as a female, but when you walk off the stage as a scientist. So anytime anyone gives you an opportunity to do something like this or to get an award, use that as a platform to talk about science. And once your science is out there, it doesn't matter, once you're talking about data, it's not about you, it's about the science. And then all of this sort of imposter stuff can just go away. So I think, so just to close, I want to just end with this one concept, which I think is what the real battle is right now. And I think this has to do with a new definition of the imposter syndrome and how Wikipedia has to change their site for 2017. So the imposter syndrome is a concept describing low-achieving individuals who are marked by an inability to internalize their lack of accomplishments and have no fear of being exposed as a fraud. So I would say don't be so negative. Don't expend so much energy worrying about where you fit in the world. There's a battle to fight, and let's fight it. Thank you.